All right, everyone. So we're going to be listening to our second plenary talk, which is going to be taking a look at the ecology of some top predators, and in particularly today we're going to take a look at killer whales, and we're going to be seeing how we can use passive acoustic monitoring to dive along them, take a look at their foraging behavior, which is quite unique, at their dialects, and try to estimate prey abundance and spatial distribution of these species. Let me introduce to you Gaëtan, which is going to be our plenary talk today, and I'm going to let him talk in just a minute, because for quick info, we're going to be taking a picture right after the talk, when the talk is over, so please don't go anywhere else and exit through this door for a quick picture. Thank you.
Okay, thank you very much. Thank you for people organizing this conference. It's really nice. I think it's a very great opportunity for young researchers to be able to gather. It's less stressful, and also because you are doing the science, so it's really great. So I will present you some of my work I've been doing before. So doing my PhD and some postdocs and master also, and so I wanted to bring you different tools of passive acoustic monitoring, focusing on one species to show like different stuff we can learn about same species, but using different tools. So. In general, but I think most of you already know, like if you're an ecologist, the interest to investigate top predator, it's because by investigating their behavior, you could have some indication, some good proxies of how the ecosystem, if you have a good ecosystem, the quality and the quantity of prey, for instance. So it's everything comes from this prey-predators relationship. So if you have a lot of predators will be able to feed, and so they would be able also to reproduce. So that's why it's also interesting to focus, and sometimes it's easier to investigate on big individuals, like a very small one, but the purpose is always to better understand how the ecosystem is going. How to observe. So I like to talk that. So if you want to use it, you are welcome to use this phrase. I like to say when we do passive acoustic monitoring, and especially underwater, how to observe what can be seen. And it's true that to monitor like top predators, so there could be big animals, but we only saw a small part of them at the surface. So if you do some ethology, you need to observe them. So we had interesting talks this morning on monkeys and primates, and so it's nice to observe them behavior. But underwater, it's much more complicated. You are dependent on the light. It's hard to know what they do at night. You are dependent on the logistical constraint. You have to be able to go there. You have to have a boat to go out on the water to find them, and this also comes from the weather dependence. If the sea is rough, you can go out. So a brief route. So what brought me? So I've been interested in. I did a lot of my work on behavioral ecology, and also on like to better understand the pressure, like anthropogenic pressure on ecosystem. And to do that, I've been using some biologging techniques, and so we have a great team. You have been able yesterday to see a presentation of Sky. So we try to understand the behavior of animals to have a good, better understanding of the ecology, and the passive acoustic, of course, which you can have long-term monitoring. So you have a broader scale, and it's an excellent tool to study in some top predators, especially predators who are vocalizing. So it's a big issue, in fact, because if you have some animals that are not vocal, it's it's harder to monitor them underwater and using passive acoustic monitoring. So that's why also I wanted to focus my talk on killer whales. We saw an interesting talk this morning about dolphins, and killer whales are the biggest biggest family of dolphins. So they are also a social. They are social. They live in a matriarchal units, which is very important for them, and it's also important in the acoustic repertoire. There have been interesting studies showing that some population have dialects, and it is. So we have learned a lot on killer whales, especially on the population studied in British Columbia, because they have been followed since the 70s. So a lot of studies and a lot of knowledge about the ecology of killer whales have been learned there, but it's mostly on the ecology of this population. And it shows there's a lot of different population, and also what we call ecotypes, which are still not really well known. And so briefly to define, an ecotypes is a group or population of individuals. So two ecotypes would belong to the same species, or at least what we think, and but they have different ecology. So this is a social species distributed worldwide. They're among different population. We can they have acoustic repertoire which is quite close. Most of population will share the same type of calls, but every population will have their own dialects, or at least acoustic repertoire. So, for instance, what we will have would be the pulse calls. So these are quite short calls, and most of them are stereotypes. So that means it's always the same, and this could define then this stereotype code will define an acoustic repertoire, and this code could be shared at like at a population scale, or at a matriarchal unit, so at a family scale. And you have also between them what we call clans, so it's different units sharing the same acoustic repertoire. And it's this thing also, if you look very close to the code, that individuals could have also their own signatures. So this will give information about their identity. You have also the whistle, which is very close to the other cousin of the dolphins. So because this is higher frequency, it is think that we thought that it could be vocalization used at close range communication, because whistle can't propagate very far because of the high frequency. And most of these whistles for killer whales are also not always stereotyped, could be more random. So there's two type of sound. The other sound, which is very interesting when you do passive acoustic monitoring, is echolocation, because as every toothed whales, they have the possibilities. Also, they have a biosonar. In fact, so they will use some sound to when they are prospecting their environment. So if they want to see in the dark, they will be able to use this. So we were talking about bats with some people this at lunch. So it's the same, and this is quite characteristic because it will be not a tonal sound, but a broadband frequency sound. Well, it's not a really good quality, sorry, but it's really sound like a click. And so what we have to understand is, like, for every click, they receive an image. And sometimes what I like to say is, like, when they are prospecting and they are not doing foraging, they will make one click. They have one image, so they are in low definition. So it's the same as movies. Now you have, when we say we are at 4K, it's like 4,000 images per second, something like that. But it's very high resolution. And so when they will go closer to a prey, they will increase the rhythm of clicks. So they will get a better resolution of the image and of their environment. So this information is important because when you are listening to them, you could have good proxies of what they are doing. Are they prospecting the environment? Are they maybe trying to catch a prey or not? I wanted to show you a study I've done with some colleagues in Scotland. So we work on the population of the Icelandic killer whales. So this belongs to the Scandinavian herring eating ecotype. So what is interesting with this ecotype is like they have particular foraging strategies. They will herd the herring in a school using a particular call called herding call. So you can hear it's quite different as there are other calls when they play with the tone. It's a monofrequency tone because the herring is sensible to this frequency. The herring, when they will hear or will detect this sound, they will react as an anti-predator reaction by gathering in a tight school, and then the killer whale just have to arrive and hit the school. So they hit, so you can also hear that, and so it gives you very good acoustic marker of their feeding activity. So every herding call, you can associate them that they try to gather the herring in a school, and then the tail slap, they hit the school. So what was interesting, it's most of the time you have the herding call, 1 second stop, and they hit. So what they do then, so there is one individual swimming quite fast. So sometimes they are not the same, those that turn around, they make the herding call. One individual arrives, debilitates the herring by slapping in the school. The fish are stoned. I don't know if this is the term in English, but the other member of the family just have to arrive and grab the fish. So the study we have conducted, we had one hydrophone deployed in a fjord in Iceland, so in a small area. This is interesting because the fjords are killer whales enter in the fjord and go back. So we had 1 month or around 1 month of recording. So the idea was to look what do they do during 1 month, and also the big question at this time was do they feed at night? So we have several types of calls. So we big categories. We annotated them manually. We have the herding call. You just listen. We have another type of herding call, which is quite funny, and you will listen. So it's a non-linear call. So just for fun, we were saying it's the teenager herding call. It could be, we don't know, but it's either we don't really have clue about what is it for. Is it like some young juvenile learning this call and they don't succeed to make this call properly yet? Or we'll see later, it could be also to have a larger variation of frequency and to frighten the herring, because the herring have, like I said, there is a sensitive at one frequency, but it could be a small variation per individual, as we are, like we have some people have better sensitivity at low frequency and the other at higher frequency. So it could be that, but we don't really know. So then we monitor the tail slap and different, so there are monophonic called and biphonic called. This is just, in fact, killer whales are able to produce 2 sounds at a time. It's just like the Mongolian thing, if you already heard that, you can produce 2 sounds at the same time. And we have the whistle, and what is called high frequency whistle, it's just because the teams there have discovered that they could produce very high frequency whistle, which is quite different. But you can't hear that, it's too high in frequency for us. So basically, we monitor the different, the mean number of calls per hour. So this is we put for all the months we gather. We look at which hour at the time do they produce this sound. So do we have more sounds at night or during the day? So the color represents day. In green, it's the twilight. So dawn and au crépuscule, sorry, and during the day and night. So basically, it's not really easy to see a high tendency, but what we observe is like it seems that there are more herding call at night than during the day, especially this non-linear herding call. And we have, what was interesting that we saw that we have a lot of taste lap. We have taste lap also at night and during day. So for the analysis, what we done, we have for every file, acoustic file, we monitor the number of calls per file. We use a statistical description to see the number. So it's the interval between files without any sound. So here in gray, you have a file without any sound. And we were able to, so this model shows that when after 10 files, you have a lot of files. The distribution is quite random. Here is an histogram, it's a distribution of intervals between files. And after a certain amount of time, you could have, it's a variable, you have a lot of, you can have either 20 minutes, 25, 35. And so we were able to determine that after 2 hours of presence, without any sound, we consider a new event. So this means because the killer whales enter the fjord, they could stay for a few minutes to a few hours, and they are leaving. So we were able to determine these units of analysis, and we were looking to the number of these events and the production rate of each calls per event and compare between day and night. So the idea was to look, do they have different behavior between day and night? So this is not straightforward, but the first thing was interesting. As I mentioned, the taste laps and the linear herding call are good proxies of feeding behavior. So we were able to see that killer whales, Icelandic killer whales, are hitting during the night. And it seems that they need more herding call at night than during the day. And this strange nonlinear herding call was mostly produced at night. So that was why we have this hypothesis that maybe it's during learning time at night and they feed more. Or it could be because, in fact, the hearing at night, the school is more dispersed, so it's not gathered. It's more during the days as they are gathered together, and at night the hearing is more dispersed, and they go close to the bottom. So we thought that they make more herding call to gather the school, the hearing in schools, that they need less this during the day, whereas the hearing is already in school. So yeah, basically it was a first example of what we have learned on the ecology of this population thanks to bioacoustics and passive acoustic monitoring. I will now present you another work. So it's a project I've been working in several years about depredation behavior. So it's the project Orca Depred, which was part of CNRS lab from University La Rochelle and the ENSTA Bretagne. Here, so I don't know if you are familiar with this depredation behavior, but I thought that a video is better than a long explanation. So these are long lines. Long lines are set at the bottom of the ocean, and it will remain a few days, a few hours to a few days at the bottom, and then a long line will be hauled by the fishermen. And when the long line is hauled, killer whales and also sperm whales in this area come and grab the fish. So this fishery is around the French. Austral and Antarctic islands, the Crozet and Kerguelen. So here it's in Crozet. It's a killer whale population of Crozet. And why did we study this interaction? As you can see, when killer whales arrive, there is no more fish really going up. There are several major issues because it could ask some questions about ecosystemic balance. Here you have a balance where we know that sperm whales feed on Patagonian toothfish and other prey. the crozet ecotype, they forage on fish, but we were not sure how much toothfish they are feeding on. They also, they are known to hunt elephant seals and penguins and other marine mammals, so they have a large dietary scale. And the depredation, in fact, behavior, they will probably switch to feed more on the boat when the boat is holding the long line. And what happens is to finish their quota, the fishing boat will spend more time at sea. So in this area, they succeed every year to finish their quota, but it requires more effort. And the difference could be quite a lot. We estimate that 30% of the quota is removed by killer whales and sperm whale in crozet. So to have an idea, in fact, in crozet they have about 1,000 tons of fish to collect every year. So they are very big fisheries. So that's why it's also a social and economic issue. It's one of the biggest fisheries in France. It's not really well known because we sell the fish in America and in Asia, but we don't eat it here. It's a very expensive fish. And to gather 1,000, so because they finish their quota, in fact, to gather 1,000 ton of fish, they have to fish 1,400 ton of fish. So there is an additional extra catch. Everything has some question about the ecosystemic balance. So if they need to fish more, and if the killer whales habituate to this fishing, to this foraging behavior, we could also disturb the ecosystemic balance, like have some interaction, like disturb the predation pressure on the toothfish stock. And it could be an impact on the conservation of this fish, and also some regulation of prey predators regulation about killer whales on this species. And like this is an example of the killer whales in crozet. They have the same behavior in Patagonia. They beach voluntarily to catch the seals, and there are only a few individuals able to knowing this behavior. So it could also be a loss of heritage, behavioral heritage. So just an example. So what we were interested is we know that 3 phases of fishing. So the setting phase, the boat sets the long line. There is a sucking phase where the fish will be caught on the long line, and then the holding phase, the boat is removing the line, and this is when we have the interaction, or at least where we can see them. So every interaction in crozet, when we estimated 30% of depredation, is because we observe the interaction during this phase. But the big question, so when I started to work on this project, was what's happening underwater? Do they interact before the holding phase? The long line was set between 500 meters and 2,500 meters. We thought that killer whales couldn't reach this depth at the beginning when I started my PhD, but there were some colleagues who deployed tags on killer whales. In fact, killer whales could reach 1,000 meters, so they are deep divers. And we were thinking, okay, maybe if we can see during this phase, we can at least try to listen if they are coming close by to the long line. And so it's what we did. We deployed several hydrophone on the long line. So the hydrophone is set for a few hours to a few days, and we record, and then we annotated, thanks to a lot of people, we annotated the presence of the killer whales, and we annotated the calls and click-off echolocation, or at least buzzes, because, as I mentioned previously, buzzes are good proxies of foraging behavior. So this is an example of acoustic recorded nearby the hydrophone when the boat was away. And if you are familiar with sperm whale, you can hear also the click of sperm whale. So yeah, it could be a big issue with when you have both killer whales and sperm whale coming. So what we have been able to discover thanks to this hydrophone. So in this graph, in fact, we plotted it's a map with a hydrophone, and this is the position of the long lines. And here it's a timeline. So the hydrophone is set. Then this is the distance between the vessel and the hydrophone. So here's the distance between the vessel and hydrophone. The vessel is setting or holding some long line. So in green they are setting the long line, and in orange they are holding the long line. And when they are holding on line here with a small triangle, it's the presence observation of killer whales nearby the boat. What is interesting here is like we hear the killer whales. Then the boat is quite far away and we don't hear anymore, but we observe them. So we thought that the killer whale was once the boat is starting to hold the long line, they are going on the long line, so on the boat, so that way we didn't hear them anymore. Here it's a 10-kilometer range, which we determine by, in fact, comparing the visual observation with acoustic detection. So what we did, we look at the probability to only observe a killer whale from the boat, and this is the distance between the boat and the long line. So between the boat and the hydrophone, and so every time the boat is holding a long line, we note: do we observe a killer whale, and do we hear killer whales on the hydrophone? So we are not completely sure they are the same, but with every recording we could estimate the probability to have either only visual detection or visual and acoustic at the same time. And what you observe is that we have a larger probability to observe them and hear them at the same time when the boat is closer to 10 kilometers from the hydrophone. It was interesting, and in fact it fit with other studies, like there was Patrick Miller in Scotland who estimated that the killer whales propagation, killer whale acoustic propagation range could reach around 10 to 11 kilometers. So this was important because we could estimate that indeed associate the sound when they are at 10 kilometers, that they are the same individuals. So here they are too far away. We can't hear them on the hydrophone. We observe only them. And here we guess that the killer whales were at least in a 10-kilometer range from the hydrophone. We can't be sure that the killer whale went on the long line with the hydrophone, but we know they are in the area. And they are vocalizing, and they are also here you have the echolocation and the buzzes. So this led us to assume that we have potential depredation from longline sucking on the seafloor. We are not really sure because they are around, but we use like the optimal foraging theory. If you have a sushi shop here on the port of Brest and you have a storm outside, it's easier to go to the sushi shop than to go fishing outside with your small harpoon and video. So this is at least like only hypothesis, but we have a lot of events in this area. A way to be able to be sure they go at the bottom and could interact with this fish would be to what we wanted to do at the beginning, to do some acoustic tracking. So, yeah, acoustic tracking. If I use an array of several hydrophones, and you estimate the difference of arrival time between your hydrophone. So it seems simple, but underwater the sound doesn't travel straight forward because the speed of sound is not constant underwater. So this is a big issue when you do acoustic propagation. So this is nice Belop propagation Sky talk yesterday about. So it could be long, but it could be very efficient, and also it shows you this is so it's over a long distance, but it shows you that you can have what we call shadow area. So that means you can be closer to the hydrophone, Or imagine here you have an individual vocalizing, you have an hydrophone here, you won't hear a thing, and here you will hear the individual. Look, it's important to understand this, and using some acoustic propagation model, so Belop take into account these effects, you can then simulate it, so make some model, and make some hypothetic propagation. You would estimate on your array the arrival time. You can estimate the angle also, because if you have very close hydrophone, you can estimate the angle by difference of time. Here you can estimate that the wave is a plan, so you can estimate the angle of arrival. Comparing the simulation and real data, you could estimate the likelihood, like the probability where the individual occurs, but so this is theory, because when reality meets theory doesn't work. Yeah, because I didn't thought that the array would be like this in the Southern Ocean, so it's hard to have it straight like this. But it's an interesting approach and could be very useful, and there are very nice study where they have been able, like in Alaska, to estimate this. This is a big table, but just we don't have to read everything, but it just summarizes every event around the long line with acoustic. What is interesting here is like we have several events where we have no visual observation when the hydrophone was old, so that means we would consider no interaction on this long line, but we had presence, acoustic presence of individuals vocalizing and acoustically active, and also maybe foraging nearby the long line in absence of the bot. So this was a big issue because it shows that we probably underestimate the depredation rate, because we have some unobserved interaction at the bottom, where the bot is not here, while we only consider the interaction rate. So this show we had to go further, and I would say like yeah, the issue might be underestimated, and we probably need more investigation on this behavior. What I didn't tell you is like we have two ecotypes there. So this one are the Crozet ecotype. It's I think close to the type A in subantarctic kilowaves. But you have also the type D. So they're very, very small eye patch. So they're quite different. It looks like an hybrid between like pilot whale and kilowatt. So it's I've never seen them. But they are quite rare. They have been described. They've been observed for a long time, but they have really been described like few years ago. And we don't really know. We know there are different ecotypes, but we don't know if there are different species or not. But they are very close to the other kilowatts. This is just Mary, the PhD of Paul Tixier, who did a lot of photo identification from the bot, and were able to estimate the interaction between the fishery and these two ecotypes. These ecotypes have a different ecology because they are thought to be offshore kilowaves, so mostly probably foraging on squid and on fish. And they are always in a large group, so when they arrive, the fishermen are really not happy because so it's interesting also maybe to better understand the two ecotypes. And we have been able to associate it thanks to photo identification at the hauling, which ecotype was present. And from this data, we focus on long line where we were sure that it could be only one of the other. Here we had the two ecotypes observed also, but at different phase. They rarely mix together. When there is one ecotype, the other one doesn't come close. So what we were thinking is like, can we acoustically discriminate them? Could be interesting to know if you only have passive acoustic monitoring to be able to estimate the interaction between an ecotype and another, and in the long term also to be able to see if there is an ecotype interacting more when the long line is at the bottom than the other one. So we don't really know yet. So what we did, we wanted to compare the call types between the between these two ecotypes. And what we had is like it's quite rare, but sometimes we have some observation of the type D in Kerguelen, and we have here one encounter where we have vocalization of this ecotype in Kerguelen. They were observed there, and we are sure it's not crozet because the killer wave crozet they don't go to Kerguelen. And what we were thinking is it could be fun to try to. We are not sure they are the same population between the two type D ecotypes, so we consider the two groups might be different, or at least they could have different vocalization. And the third group is the crozet ecotype. So yeah, so we wanted to make at least do we have some code differences between the ecotypes and different sites. So it's something we saw this morning. We did some manual annotation. We annotated the fundamental and the first harmonic, and made a lot of estimation of features. There is a paper we are preparing, and Salome, so she was a student working on this, and she makes some very nice analysis. I didn't show you, but could discuss. We compared between different annotators because this take a lot of time. So we had several annotators, or 3 annotators who make different annotation, and we make an interesting analysis of annotator variability. And what we observed is like according to the metrics, we have a few metrics different between annotators. So at least we take a sample of all the annotation, and all the annotation were annotated by 3 different annotators, and we compared how different it was between them. And what we obtained is like there are 5 metrics features that we can be almost certain that the variability between 2 annotations are not due to the annotator, but probably due to the vocalization. So this is a huge, we were talking about annotation, and this is a huge thing, and there's a lot of work in Bretagne where we had a lot of discussions there, because it could introduce large BIA, annotation, manual annotation by annotator is interesting, but everyone doesn't see the same thing, so it could introduce large BIA. And so this approach was really interesting because we were able to be sure about yes, it's more robust to say, okay, if you have differences between some metrics, it's more likely due to the calls and the annotation. And what we observe is like we have differences. So in blue, there are the type D, so in crozet and Kerguelen. So we don't really see a lot of variation between this box plot. In fact, in crozet, we have several encounters of type D annotated here. But in Kerguelen, we have only one encounter, so it must be the same individual. That's why the distribution is not as large as the other one. And in crozet, we have several encounters with a lot of different, probably, families. But we had some significant differences between the crozet and the type D. And for instance, what we have been able to observe, like the crozet have a shorter vocalization, but make sound at higher frequency, like it's. Yeah. So another method we wanted to do is we use a linear discriminant analysis. So it's quite an interesting approach. This kind of machine learning approach. And what is interesting is like we were able to compare the 3 groups using this approach. It gives you this kind of graph. So you compare between the different. It's kind of a ACP, a PCA, and you have the distribution of the values. So we can see the crozet type here, the type D, and the type D from Kerguelen overlaps the distribution of the points of the type D from crozet. So this gives us information that using these features we can discriminate. It seems that we can discriminate both ecotypes. we have indeed quite good, it's not perfect, but we have quite good accuracy, but the small amount, I think it's come from because we separate type D from Crozet from the type D from Kerguelen, and we can see, in fact, so this is the prediction of the model. It will associate the type D from Crozet to 68% of the actual type D, but the Kerguelen, sometimes they mistake and they associate them to the type D from Crozet, but the Crozet types, they rarely associate them to the type D. So it's quite interesting. And what we did is, like, if we didn't know, so we take a study case, we didn't know these were individuals observed in Kerguelen, they were type D. If we only had acoustic, would we be able to associate them to one or the other ecotype? So we learned the model on the vocalization from Crozet. So this gives you the results. And what's interesting with the linear discriminant analysis, you can learn the model with, so it's a supervised approach, but then you can apply it on new data. So this data tested, we knew the result, but we had a dummy approach saying we don't know. And what was interesting, like they associated these individuals at 83% to the type D from Crozet. So individuals are. So I thought it was really nice, this approach, because it shows that we could have a certain amount of we could be sure at 80%, but it shows that if we have a new recording in this area, if we don't really need the visual confirmation to estimate if it's one ecotype or another. So also this gives you, so this is on one event, so if you have this percentage, you can probably conclude that it is in fact it is indeed type D kiloways. So yeah, so a really interesting approach, and yeah, I've been a little bit fast, but thank you for your attention.
Does anyone have any questions for Gaëtan?
Thank you for the talk. Is there any solution to avoid depredation? Like,
yeah, stop fishing for once. No, it's not a good answer, but it's really complicated, and that's why also we wanted to know if the killer whales interact at the bottom. So all this research on the Orca project was to better understand the behavior of these individuals, of this population of killer whales and sperm whales, to maybe try to find some solution. Because at the beginning we were thinking you could find some device protecting the fish when the long line is old. So it's, for instance, in Chile, they have this, they call the cachalotera. It's like big socks, floating sock. And when the long line are old, the hooks are protected by this sock. So it seemed efficient to avoid sperm whales to interact. It decreased the interaction in this area. But what we saw here is like we would probably move the issue, and they would be able to interact on the long line. So I didn't show everything, but we had some interesting study also on sperm whales in this area, where we were sure we had some real interaction with the long line at the bottom, because we equipped the long line with accelerometer, so we have a movement of the long line, and we saw like the long line moving for 20 to 30 meters up, and the next day there were a lot of sperm whales everywhere. So it's very important to understand this to find a solution. But if you want to find a technical solution, so to protect the fish, a solution would be to think the fishing process. So maybe by using fishing pots. They tried this. It seems efficient, but there is always this trade-off about is not this fishing changing the fishing way would be less efficient than the long line. The long line also have the advantage that is more selective. They don't have a lot of other species caught on than the targeted species, which is a Patagonian toothfish. But yeah, it's a very big issue. In Alaska, they try to use some acoustic decoys, so they put some hydrophone who can play back some sound. They put some dummy long lines with this, and they put the sound of a fishing boat holding the line, and so sperm whales are attracted by this sound. And that was also one part of my study on this. It was to listen to the sound of the fishing activity to understand what the animals can hear. And what aspect? Well, I can show you briefly. This is the acoustic of a boat going backward. So sometimes the long line is stuck on the bottom. So they make some maneuvers, and if they go backwards, they increase the sound. So this is something also we can advise them. Give some advice to say, okay, if you drive smoothly, you will decrease your detectability. And we estimated that it can propagate 3 to 5 times longer than regular hauling activity. So we called it like it could be as a dinner bell, effect. So say to killer whales at far distance, okay, we are hauling a long line. So there are a lot of also techniques. Maybe they can play in techniques. It's what they do in Crozet. They have a different fishing strategies than in Kerguelen. Kerguelen, they are only sperm whales, and in Crozet, they have sperm whales and killer whales. The array is smaller, so they will put smaller long lines. They will move at a further distance. So they have a really different behavior. And this is the acoustic signature of the setting. So we show that the killer whales and sperm whales are able to know when and where a long line is set because there is a different acoustic signature. But this is very difficult to play on, or maybe to change the way to deploy. But yeah, it's really tricky. There are also in species who can learn very fast and adapt that behavior, their learning behavior, and it's not an easy subject, but. Yeah, and another way would be maybe to help fishermen to know where the killer whales are, or to monitor the area by using passive acoustics, so they could hear. A fisherman can hear if there are individuals in the area by acoustic. Could be a way to make them to help them to make good decision, because most of the time when they decide to change their behavior, when they see a killer whale or sperm whale, it's too late. It's because they are interacting with the long line. So. Very tricky question.
Anyone else?
Is there a way to monitor or to estimate the quantity of fish that are took by the killer whales in the deep sea?
In the deep sea, no, not really. I don't know if I have here. No, but in fact, what is done is you compare the catch per unit effort, so it's the number of fish caught per long line. So you have a quantity in a kilogram per hook, and you compare when you have interaction at the surface, and when you don't have interaction at the surface, you compare your fishing efficiency. But so the question is like how to monitor this is really complicated, because what we thought is like if we have some, we thought that so the main depredation we are almost sure it's during this phase, but what we observe is like if we have some interaction at the bottom, the catch per unit effort on this long line considered as no depredated will be lower than it should be. And it's really complicated because when the long line is set, so it's there are very big long lines. They could these long lines, they are between 3 kilometers to 10 or 12 kilometers. So they have automatic baiting. So the hook is like 3 baits per second. So and not every hook has a bait, so you can estimate the number of bait at the end, you know. And so this is one issue, and also sometimes the bait are eaten by other species. And no, it's very, very complicated. But this method works quite well to compare the catch per unit efforts. But we think we probably underestimate a little bit the catch per unit efforts. Yeah.
Thank you.
Or maybe no, I don't know.
All right. Well, thank you, Gaëtan.
Thank you very much.
Talk. And so, yeah, if we can just meet outside for 5 minutes before coffee, that'll be great. Thank you.
All right, folks, let's reconvene. We're starting the next session, the best session, of course. It's going to be a quite long one. We have 5 presenters. The title of the session is Listen Up, The Secrets of Acoustic Monitoring. Our first presenter is going to be Peyret. Did I pronounce your name right? Okay, thank you. And the title of his presentation is Uncovering Overseas Migration Patterns, a Computational Bioacoustic Approach.
Thank you. So yes, I'm Peyret, and I'm going to talk about bird migration and its monitoring with passive acoustic monitoring. So first, a few words about Biophonia. We're a consultancy company. We're specialized in bioacoustics and applied bioacoustics. And we're supporting biodiversity manager with innovative team tools. We're using machine learning and passive acoustic monitoring to collect the data on biodiversity. And we take care of the acquisition, the storage, and the analysis of the data. We work for biodiversity managers, parks and reserves, local authorities, private companies and associations. We also work with labs, and we have collaboration with labs when for some research project. And I'm going to present one of these. So let's talk about bird migrations. It's a project that we lead with the National Museum of Natural History. It's called Migrate Lane. The goal of this project is to understand the migration patterns of birds above the sea on the western French coasts. It's a big project, multimodal project. So there is passive acoustic monitoring, there is radars, and also visual observation to understand better these patterns. And our work package is to study through bioacoustics and passive acoustic monitoring. So the context is that there are lots of wind farms that are under project, and some are already operational on the western coast of France. And there are some potential impacts on birds, and mostly on migrating birds, because those are migrating routes along the coast. And the behaviors of the birds are either avoidance or attraction, and this is sometimes leading to collisions. So this is a problem for those migrating bird species. And the goal of the project is to understand what is the behavior and what are the migrating patterns of these birds over the sea and on the coast, and especially in this area. So guess what? We're using passive acoustic monitoring, and we have recorders that have been deployed by the museum all around the coasts. Last year it was 15, and now it would be more, I think, more than 50 recorders in the project. here's an example of one recording in Wesson Lighthouse. They're installed on private buildings, boats and ferries, natural reserves, wind farms and lighthouses. And so the data will be crossed with visual observations and radars. So to do the analysis of the bird migration, we use passive acoustic monitoring, but we cannot annotate the data manually, so we use deep learning, and we want to process those data automatically. So the first question is, which model can we use to detect automatically? And after it would be, how can we improve this? But first, let's see what's available. We have one model, which is NBM model. It's a model built by citizen science project, which aim to detect nocturnal flight calls. It's citizen science, so anyone can join. There's the BirdNet, of course, and there is the Google Perch model. So my question is, which one is the best to use for our use case for detecting migrating birds? So first we want to create a data set of annotated bird calls to compare the models. And so we had a lot of recordings, and we have to sort out those recordings to build an evaluation data set. So for that, we did pre-detection with BirdNet. We know that it may include some bias because we use pre-detection from BirdNet, but we don't really have any other option because otherwise we would have to listen to days and days of recordings. So we predicted some events with BirdNet, and then we extracted 1 minute of recordings around the detection. We stratified. We used stratified sampling by species, hours, and sites to get something evenly distributed, and then we led an annotation campaign. My great colleague Tanguy Lois did this annotation work, which was a huge work. He annotated 11 hours of recordings with his expertise, and he drew boxes around sound events on spectrograms using Audacity. So we ended up with more than 5,000 manually boxed annotations, and we detected, or we confirmed, 93 species. But for some species, we didn't have enough annotations for our evaluation dataset, so we kept only 38 species for which we have at least 30 boxes. So let's go back to our main question. What's the best model available today for this task? The problem is that those models have different tasks. So some are doing event multi-class classification, and some are doing segment-based multi-level classification, which means some are drawing boxes around events, and some are just checking if in one segment there is a presence of one given species. So NBM is event-based multi-class, and BirdNet and Perch are segment-based multi-level. So that's why they have different label segment size, and they also have different model backbone. That's detection transformer, and for BirdNet and Perch, it's EfficientNet, two different versions of EfficientNet. So to homogenize all this, we had to map the detections on a regular time grid. We chose 5-second time grid, which was the largest grid of all models. And so we had to use 1 second overlap windows with bird nets to align with the 5-second segments of perch. And for NBM, we just mapped the detection on the 5-second window. Then we considered that a detection was a true detection when the confidence score was above 0.5. Then this is led to this ugly table, but it's just to say that we got precision, recall, and F1 score for every one of the three models, and we got these scores for all the 38 species that we studied. But let me sum this up for you. So these are the recall, precision, and F1 score of the models. And the conclusion for now, it's really fresh data, so we need to dig a bit more into the data and see what's all the performance by class, and so by species. But the NBM model is performing poorly compared to the other ones. BirdNet and Perch are almost equally performing. So that's a first picture of the performance of the different model existing for this task of detecting migrating birds on the western part of France, western coast. And there is room for improvement, so that's one first conclusion. And we can comment this. Maybe NBM is a small project, and the database for training is really smaller compared to BirdNet and Perch. So that may also explain the lower precision and recall. But we want to move forward, and so our next goal will be to train new models to improve performance. Maybe we'll use transfer learning or go into new models from scratch. Then we will analyze data over 3 years and more than 50 sites, and then we will cross all this data and study spatial temporal patterns based on the species detections, and hopefully that can help to better understand the migration patterns over the sea in the western coast of France. This is Bioacousticians team. Just one quick comment is that we are based in Corsica, but also in Saint-Étienne, and don't hesitate to reach us if you want to know more about Applied Bioacoustics. And thank you for listening. If you have any questions.
Yes, thank you.
Thank you for your presentation. You mentioned you have microphone all around France. Do you know what's the detection range of your microphones, or is it something not communicated?
It's a very good question. Thank you for asking it. The detection range of the microphone is a tricky question. Actually, there is no clear answer, but we have one PhD student, Manon Ducretet, who's working on this right now. She's studying, she's building a probabilistic model to predict the area of detection around the recorder. And this depends on many variables, as you may think. We have, in this particular case, a very noisy environment. Also, when it gets windy and you have a microphone on top of wind turbines, you can the detection range could be lower. It's something to work on. We know it.
Okay. Well, thank you.
Any other questions?
So it's not really about the models, it's just you mentioned that the Migratland project intends to work on collision, and I'm wondering how you intend to do it.
I was exposing the risk for the birds. Migratland project's goals are not especially studying collision, but more the behaviors of birds when they're over the sea, when they're migrating. We don't know at which altitude they're flying. We don't really have a clear picture of which species are far away in the sea, or which species are more close to the coast. And having this information will provide more insights for the wind farms operators to mitigate the impact.
Okay.
And the risk. Yeah.
Thank you, Paul.
Thank you.
Thank you. Thank you.
Our next presenter is Arienne Calonge. Did I butcher your name? No. Okay. Her presentation is the importance of multi-sensor observations to advance species co-occurrence knowledge, a demonstration of two acoustic technologies.
Good afternoon, everyone. I'm Arienne, and on behalf of my colleagues, Yulene, Carlota, Jan, and Elizabeth, I would like to talk about the importance of multi-sensor observations to advance species co-occurrence knowledge, a demonstration of two acoustic technologies. So that was quite a lot of words, so we're going to break it down. So first, spatiotemporal co-occurrence. It's a common knowledge in ecology that species co-occurrence is vital if we want to maintain community structure and function. And so if we want to protect the ecosystem as a whole, we must take into account species co-occurrence when investigating species. And so in this study, we investigated the co-occurrence of 4 species or species groups. So first, the European sea bass and the Atlantic cod, so both of which are very heavily overfished in the North Sea. The sea bass has been declining since 2010, and the Atlantic cod has gone through several fish stock collapses in the past. And then for the cetaceans, we have the harbor porpoise, which is the most common cetacean in the Belgian part of the North Sea. And then we have the white-beaked dolphin, and together with the harbor porpoise, they are residents in the Belgian part of the North Sea. And then we have the common bottlenose dolphin, which is considered a regular visitor. And so these cetaceans are actually subject to a lot of anthropogenic activities, as you maybe you don't know, but the Belgian part of the North Sea is one of the busiest seas. We have a lot of shipping activity, fisheries, and also offshore wind farms. Fisheries bycatch alone, it's already one of the primary causes of the decline of marine megafauna. And so we're using acoustic technologies to inquire into the co-occurrence of these species or species groups. So we have passive acoustic monitoring and acoustic telemetry. So this is a CPOD logger. We all know that cetaceans echolocate, or they produce sounds to navigate or forage. And with these CPOD loggers, we can actually study the presence or absence of these cetaceans and even their behavior over a certain area. So these are click trains. The one on top is an example of a harbor porpoise click train, which is known to be narrowband high-frequency click trains. And then the one below, these are broadband click trains of dolphins. And then for acoustic telemetry, these fish animals are surgically implanted with acoustic tags. And these acoustic tags transmit acoustic signals, which are then detected by these receivers. And so is it actually feasible to use passive acoustic monitoring in combination with acoustic telemetry to inquire into their spatiotemporal co-occurrence? And why are we even using these two techniques in the first place? Well, it was quite opportunistic because in the Belgian part of the North Sea, we already have a cetacean passive acoustic network that has been logging the presence of porpoises and dolphins since 2018. And these seapod loggers are fitted into a multi-use platform, as you can see here, and it's with a tripod frame design. And this multi-use platform can also be fitted with an acoustic receiver with an acoustic release system, and it is part of the fish acoustic receiver network. And as you can see here, this is the cetacean passive acoustic network of Belgium. And so we decided to use the units of detection positive hour in this study, wherein a value of 1 would correspond to an observed presence of an animal within any given hour, and then a 0 would be just the absence. And then we gave a value of NA every time it was impossible to detect the species because of sensor failure or battery failure. And so we created a presence-absence matrix for every hour and for every station in the Belgian part of the North Sea. And so we wanted to distinguish 2 terms in this study. One is occupancy, which is the observed presence or detection of 2 or more species or species group within the detection range of a station in the same season. In contrast to co-occurrence, which is the observed presence or detection of 2 or more species or species group within the detection range of a station at the same hour of the same day. And so in our study, we did 5 different co-occurrence analysis, but then in this presentation, I will only be showing you 3 due to time constraints. So first is the co-occurrence percentage, which is an hourly co-occurrence, and is basically the percentage of the animal's solitary hours and its co-occurrence hours with the other animals. And so in this table, I would like to highlight the high co-occurrence percentage of cod, sea bass, and dolphins with the harbor porpoise, which ranges from about 30 to almost 50% of the time that they were detected. And so we decided to focus on these 3 species or species group pairs in the subsequent analysis. We analyzed their diel overlap pattern, which is the extent of seasonal diel overlap. And the gray area, the shaded gray area, is basically representing the diel overlap coefficient of the species pairs. And you could see high diel overlap coefficients for the fish and the porpoises. And this could be just attributable to their patterns of occupancy. So as you can see here, the fish were rather detected continuously throughout the day, while the porpoises were mainly vocalizing at night. And then lastly, we also did co-occurrence modeling and a temporal resolution of an hourly co-occurrence. And it's basically asking the question that when a sea bass is present, what is the probability that it is co-occurring with a harbor porpoise, given that, of course, the PAM logger is active within the vicinity of the station. And so using a backward selection model, the best fit model was a GLM model with a diel and season as factors of predicting co-occurrence. And so significant results showed that there is a higher probability of co-occurrence or detecting a sea bass that when a harbor porpoise is there during the night and during the colder seasons, which is during autumn and winter. And so we tried to look into different literature and to explain the reasons for these co-occurrence patterns that we're seeing. And the obvious reason is diet. And so cod and sea bass were found to be 2 of the prey of bycatch porpoises from the Belgian coast. There were reported aggressive interactions of dolphins and porpoises, and also lower co-occurrences of porpoises and fish during the summer is actually coherent with reports that there is a shorter supply of quality prey during the summer, hence a lower blubber thickness and a higher risk of starvation for these porpoises. However, these fish are also top predators in the Belgian part of the North Sea, as well as the cetaceans. The tagged fish are also way out of the size range of the average prey size length of porpoises based on stomach content analysis. So the fish that we tagged are between 33 to 66 centimeters, while the average prey size of a porpoise based on stomach content analysis is only between 2 to 15 centimeters. And so there's also been studies that were found that there are actually dietary overlap among these top predators over mackerel, scad, sardine, anchovies. And so they're all opportunistic feeders as well, and that it's a good point that porpoise distribution actually highly reflects the distribution of its prey. And then the higher co-occurrences that we found during the night is also coherent with some studies that we found that says that porpoises actually forage in deeper waters at night. And during the colder seasons, these endothermic porpoises actually require a higher food consumption to be able to regulate their body temperature. And so could it be that their diet is bringing them community interactions and shared habitat preferences, could be bringing the species together and be the reason for their geographic overlap? Well, that is our hypothesis, and it's not very certain because we only investigated presence-absence data in this study and not their behaviors. But what is the big value of this research? This is the first study to actually combine these two different acoustic techniques to be able to study species co-occurrence. And it is valuable because of the long-term data sets that we're using and the generation of high-resolution species co-occurrence data. So normally, a species co-occurrence studies uses a species-by-site matrix lacking the temporal aspect, but in this study, we were able to use both spatial and temporal co-occurrence. And also, of course, there is a potential of observing more co-occurrences with the progression of fish tagging. And so to summarize and conclude, multi-species co-occurrence research is a huge added value to the data that we acquire from present technologies. Usually these technologies are being used to inquire into single species behaviors and presence, whereas in this one we are actually trying to combine them to look into species co-occurrence. And then co-occurrence patterns differ at varying spatiotemporal scales, which is why long-term high temporal resolution data on species co-occurrence is important. We didn't find any significant results when we looked into seasonal co-occurrences, but with the hourly co-occurrences, we were able to see significant patterns. And then lastly, species co-occurrence research is a step towards an ecosystem-based management of our oceans. And so as a step forward, the Marine Observation Center of LIST, because of the success of the tripod network, we are advancing into this new type of mooring that could fit more sensors. So it can fit a CPOD, an acoustic receiver, an echo sounder, hydrophones, plankton imaging, ADCP, and scanning sonar. And we are moving towards the goal of combining more data sets to further look into our ecosystems. And so thank you for your attention. If you want to check out our published paper, it got published this January, so you can scan the QR code or you can ask me about it. And then also I'd like to acknowledge that a lot of hands contributed to the maintenance of the sensor networks. It's a big effort of the entire Marine Observation Center of Liz. So I would like to acknowledge them and also the infrastructure department, and of course the crew of RV, Simon, Steven for their continuous involvement and support at sea. And I would just also like to let you know that all of our data is open data. You can publicly access them, our long-term data sets on fish, cetaceans, plankton, everything. So if you would like to get some of our data, you're welcome. You could send me a message or I will show you also how to, but you can also just Google it on VLIS datasets, and you will see everything that the datasets we're producing. So thank you for your attention.
That was a great talk. Any questions for Arienne?
This is tall. So it's okay. So now that you're getting actual hydrophones, are you going to move away from doing telemetry as much and try to record the Atlantic cod themselves and the sea bass? Because they're pretty high in amplitude for fish if you can get that low frequency.
Yeah, that's a good point. But the fish tagging is gonna continue the fish tagging work because it's very certain you can. I think it serves very different purposes. Mm-hmm. With acoustic telemetry and fish tagging, you actually follow the fish track and just their migration and all of those things that you cannot study with just hydrophones. But we're also doing hydrophones, and it's still a bit. I think we're more advanced in the acoustic telemetry part. Mm-hmm. With the hydrophones. We're working on it. But then it's kind of difficult because the Belgian part of the North Sea is very noisy. So it's not as easy to detect the sounds of these fish. Yeah, so we have a lot of unknown sounds. Maybe you saw the poster of Julia. It's about the unknown sounds that we have in the Belgian part of the North Sea. So we are trying to work with hydrophones as well to detect fish, but there's still a lot of work to be done in that area.
Makes sense.
Yeah.
Any other questions? I have one.
Yes.
So is the system that you deploy cabled, or if not, how long can it be down there?
Yeah, it's completely autonomous, not cabled at all. So we usually maintain it every 4 months to collect the data, replace batteries, make sure that everything is working.
Is the acoustic data collected duty cycle, or is it continuous?
It's continuous, yeah.
Okay.
Everything is continuous. So, yeah.
All right. Thank you.
Yeah, you're welcome.
Okay, our next presenter is Patrick Kagerer. Thank you. And his presentation is going to be passive acoustic monitoring provides a first insight of the behavior of sperm whales off eastern Iceland.
Okay, so hello everybody. Thanks to the organizer for this really nice conference. We started talking a bit about sperm whale acoustic this afternoon. Now we will dive a bit more in the subject without unfortunate pun. So sperm whales are a widely distributed species that you can find in every deep water of the global ocean, and that is most often associated with large bathymetric features such as submarine canyons and ridges. And most importantly, it's a latitudinally segregated species, which means that the females and their babies, known as calves, are mostly staying in central areas of the ocean. In the blue on the map, and the males are mostly staying in high latitude areas in light blue on the map. The problem is that males need to migrate to reproduce, which makes sense, but despite more than 50 years of research on sperm whales, we still have a very limited understanding of this migrating behavior. You can see on the map arrows summarizing what I found in the literature, and bear in mind that some of these arrows are just the results of one GPS tag or one mark recapture from the whaling era. So we really have a very limited understanding of this behavior. And you could say that it's a fairly easy question to address because you can simply deploy satellite tags and it will give you an answer. But the problem with satellite tags is that they are unreliable as they rarely last for the whole duration of the migration, and at the same time they are insufficient because we estimate that there is 840,000 animals worldwide, and if you get 100 tracks, which would already be a high number of tracks, you would still have a very limited picture at the population scale. So we need a complementary strategy, and what we could do is that we could set up an observation platform in an area of likely ecological importance for sperm whale. And as these areas are likely to be far offshore, passive acoustic monitorings immediately become very interesting because you don't have to expend a lot of resources to maintain human presence in the area. You can just send humans once to deploy an hydrophone array and a second time to recover your hydrophone array, your recorders. So that's basically what we did. We deployed a 2 hydrophone array in the Gridau Gorge, so just here. It's a submarine canyon between the Azores, a known breeding ground, and northern Norway, a known feeding ground, which, cherry on top of the cake, we have evidences of movement from the Azores to northern Norway. So the Gridau Gorge is likely to be an area of ecological importance for sperm whale. The next question is, how do you localize a whale using only recordings, and that's where it's getting interesting. What we did is that we used the depth, so depth and slant range or direct distance between the hydrophone and the whale, as well as the angle between the hydrophone and the whale to calculate a location. Calculating the angle is pretty simple. You can see that the blue track is shorter than the red track, so the sound is arriving at hydrophone 1 before hydrophone 2. Which means that using time differences of arrival between the two hydrophones, you can infer the angle. So that's a pretty simple feature that is implemented in the PalmGuard software. I won't talk too much about it. Now, if you want to calculate the slant range and the depth, you have to be a bit more sneaky because we only have two hydrophones. And what we did is that we used multipath ranging, which is a methodology that relies on the time differences of arrival between the direct path, here in dark, and the bottom reflected path, here in red, and the direct path and the surface reflected path, here in blue. You can see that these two time differences of arrival are included in this set of equations that are relating the distance, the slant range, to the time differences of arrival, as well as the depth of the water column, the source, and the receiver. So if you turn this set of equation in a matrix format and reorganize it, you get this matrix equation where you can see that the depth of the source and the slant range have been isolated from the other known variables. So in theory, it may be simple. You just have to replace variables, and you get the depth and slant range. In practice, it was a complex workflow with a high potential for optimization, since it required a lot of manual work in steps 1, 2, and 3. And while manual work is not a bad thing in itself, it's problematic because it consumes a lot of resources and also tend to reduce the precision of our position. So we only could use the median horizontal position over our 5 minutes recording instead of the position we calculated for every click. The depth, though, were not as affected by imprecision as the horizontal positions. So nevertheless, we have been able to calculate 28 tracks, so to follow 28 whales, and the median speed of all the tracks was of 1.6 meters per second, which is a good value because it's consistent with other studies that were using GPS or accelerometers. So while not being the most precise methodology we used, we still had some reliable results. And you can see all the tracks on this map. And what is immediately visible is that you cannot interpret it in this way. So what we did is that we calculate the angle relative to the north of every segment of a track. So this is the light yellow track. This is one segment. We calculated the angle relative to the north of this segment, this one, this one, this one, this one, calculated the average, and did that for every track we calculated. And that gave us this plot where you can see that most of the tracks are heading west or southwest, which is a bit surprising when you think about it, because Iceland is southwest of northern Norway. So you would expect sperm whales to go northeast, not southwest in our case. But that's not the case. But what is reassuring is that it's following the local bathymetry, which might hint on foraging behavior in the area. You can also see that the variance is greater for the southbound tracks, and in this case, variance is a proxy for reliability of the track, because you expect the sperm whales to have fairly straight tracks, and variance is increasing with the wiggliness of the track. So southwards tracks should be considered with caution. If we now look at the dives that we calculated, we can see that most of them were shallow, with 75% of them were, being shallower than 400 meters. 400 meters is shallow for a sperm whale. It's not always the case, but it's good to mention it. And you can also see that there is a slight increase between 500 and 600 meters of the number of dives performed. So if we now look at the buzzes, we can see that the buzzes being vocalization produced by sperm whales when they are attempting to capture prey. Think about them as short-range biosonars. And so we can see that these buzzes were produced all across the depth of the dive of our sperm whales, but most often between 500 and 600 meters. And that's interesting to relate to the dives, because while we have 75% of our buzzes that 75% of our dives that are shallower than 400 meters, we can see that 50% of our buzzes have been produced deeper than 400 meters, which may hint on bimodal behavior of sperm whale diving behavior, with shallow dive that may be an all-purpose diving behavior where the sperm whales are either moving or maybe also occasionally or opportunistically feeding when there is low-hanging fruit. Whereas the deep dives would be mostly aimed at foraging, because the whale has identified prey of with a high energetical content. So, if we conclude, we can say that this work has demonstrated the feasibility of offshore passive acoustic monitoring and multipath ranging and their usage for marine mammalogy, that the track that we calculated followed the local bathymetry rather than the expected migration heading, that the recording of buzzes are evidences of foraging behavior, and the association of these buzzes with deep dive may indicate a bimodal foraging strategy. Thank you for your attention.
Thank you, Patrick. Well, you know the drill by now. Any questions?
Thank you very much. A really nice presentation, and it's really nice to see some tracking. I have a question. Maybe I missed it, but how do you have the heading direction? Because on the, you show a graph and you show that you can localize in 2 dimensions, like distance and range. But how can you?
Well, basically, what we did is that after calculating the localization of the whales, we calculated the 2 solutions of the equation, and we created an algorithm based on the spread of our points, and the points that were having the less spread and that were creating the most consistent track were kept to create a track. We could talk more about it because the methodology is very long, and what I've presented here is a very simplified version of it. But yeah, it's basically an algorithm, very simple algorithm. If it's smaller than this, and if it's if it's the shorter solution, we will keep this one.
Yeah, and okay, great. And because I was wondering also if you have the distance between your, like, once you have the tracks, you may have a distance between the whale and the hydrophone. So have you been able to look, or is there anything you can draw with make a relation between the receive level of the click, the intensity? Is it possible? Because we know that they may vary. I think sperm whale is quite consistent on the intensity, but it could be a much faster solution, to consider the intensity of the receive level and estimate, like, with a log propagation. I don't know if it would be working, but
well, we don't have much resources to expand on this project, and I'm basically working on it. But I'm a biologist. I have a biologist background, so it's a bit difficult for me to go in complex acoustic equation. It may be interesting to look at it, but yeah, I don't feel that we have I have the skill to do it, and I don't think we have the resources to go in this direction.
Yeah, but we can talk later. It would be much easier than your method.
Yeah.
We can talk at the coffee.
Any other questions? No? I do have one.
Okay.
how deep, so you mentioned that you use PEM.
Yeah.
How deep on the column of water do you place the hydrophones?
At the bottom of the ocean, so the depth of the ocean is approximately 2,330 meters, and the hydrophone is so the anchor is at the bottom, and we have a 40-meter line between the anchor and the hydrophone array, which, if we talk about it in during the coffee break, will have its importance as well.
Okay.
Because there is reflection phenomena that have to be taken in account. So.
I would imagine.
Yeah.
Okay. All right. There is no more questions. Thank you, Patrick.
Thank you.
Our next presenter is May Norwood. And her presentation is going to be passive acoustic monitoring shows seasonal and dial variation in delphinid whistles off the coast of Dakar, Senegal, in West Africa.
Hello everyone, my name's May. I'm here from the University of St Andrews. This was my undergraduate dissertation project, supervised by Dr. Julie Oswald, and the data were provided by Dr. Salvatore Cerchio and Dr. Danielle Chiloyak. So, just to start off, I want to give you a bit of an overview of what I'm going to be talking about in this presentation. So, kind of talk about delphinids in West Africa more generally and kind of place it in context a bit. Then I'm going to very quickly run you through my methods and study area, and then I want to kind of go over some of the results I had with seasonal variation and dial variation, and then see what these results could maybe indicate and potential future directions following on from this. So, I looked at West Africa. The coastal countries are highlighted in green on this map. So it's a diverse region. As part of this project, I did a literature review and found 34 species were found in the literature. They're important in many cultures. However, kind of after colonialism, some of the more traditional beliefs where cetaceans are kind of venerated in some places, kind of being eroded. And again, after colonialism, there's more issues with food insecurity and poverty and things like that. So there are some conservation issues arising. So poor regulation of fisheries means there are quite high rates of bycatch. There's also directed takes, either for food, so use as marine bushmeat, or also sale to the traditional medicine industry, and also live capture for sale to the live display industry as well. There's also habitat destruction, particularly in coastal environments. So there are a lot of mangrove forests in Senegal, and these are really important habitats for species like the Atlantic humpback dolphin, which is endemic to the region and critically endangered. And this is unfortunately, this degradation is contributing to their endangerment. So there is a need for conservation, but there's little dedicated cetacean research in the area because, again, after colonialism, there's lack of funding for these sorts of things, and also kind of lack of funding at universities means it's harder for people to get skills necessary to do this kind of research, even if they are interested in it. This means that the majority of data comes from incidental sources, so bycatch, opportunistic sightings, and stranding data as well. And as I'm sure you're all aware, there can be problems with bias with these types of data. So passive acoustic monitoring offers a really useful opportunity because it's relatively low cost, so it can be employed in an area like this where there isn't much funding. So as I said before, hydrophone data were collected, looking at delphinid vocalizations. So these include clicks, burst pulses, and whistles. I focused on whistles in my study. You can see here, this is a whistle on a spectrogram. It's a tonal signal. I chose to focus on whistles because they're quite easy to identify and they are associated with some interesting behaviors like communication, socialization, that sort of thing. There was a lot of data, so I used an automatic whistle and moan detector in PAMGuard to go through it and then cross-checked a subsample visually myself in Raven Light to check that there weren't any detections being missed. One problem with the automatic detector is that it often fragments or only detects partial whistle time frequency contours. So this means you can't really do analysis on an individual whistle level. So I divided it up into whistling encounters, which I defined as a bout of whistling separated by at least 30 minutes from another bout. This is just showing in more detail where the hydrophones were placed. Up here shows the site on the coast of West Africa, and then zoomed in, this is where the hydrophone was placed. It was about 18 kilometers offshore. It recorded for 2 years, from June 2021 to May 2023. The water was approximately 320 meters deep, and this was originally a baleen whale research project, so it recorded at a sampling rate of 48 kilohertz, which is actually quite low for delphinids. That was another reason why I chose to look at whistles, because it wasn't a high enough sampling rate to really get a representative picture of clicks and things like that. So what did I find? Well, first of all, I found there was seasonal variation in encounter duration and intensity. Duration's pretty self-explanatory, how long an encounter was. Intensity I defined as number of whistles per minute, so that's kind of an indication of how often dolphins are whistling in an encounter, potentially if there are more individuals, that sort of thing. And I found seasonal variation. So these are box plots, and then these red squares represent the mean values for each month. And we found that for duration, they were significantly longer in between June and September, and for intensity, they were significantly more intense in between June and August, so during the summer, basically. There was also seasonal variation in frequency variables. So one thing the automatic detector does is for each detection, it records the minimum and maximum frequency, and I took a mean value of that for each encounter, and then looked at that variation over time. So for both minimum and maximum frequency, they were significantly, significantly lower in the spring months between March and May. So again, seeing some clear seasonal variation there. These variables are linked. I wanted to see if they were relationships, so I divided encounters into short, medium, and long encounters. Short encounters were less than 500 minutes, medium 500 to 1,000, and long 1,000 or more minutes. And we found that there was a lot more variation in the short encounters. You can see this box plot is much wider, and the long encounters also had a significantly higher frequency. And had much less variation. This could potentially be due to the fact that lots of different species vocalize with these short encounters, so there's a much more variation there, whereas maybe just one or two species vocalize with these long, very long encounters, so you get less variation. But I didn't just look at these variables on a seasonal scale, I also looked on a dial scale. So I divided the day up into dawn, day, dusk, and night, and if anyone's interested to know how I did that, I'd be happy to talk about it later. And found that frequencies were significantly higher at dusk and night than in dawn and during the day, and there was also less variation at dusk. This could potentially be due to the fact that there were fewer encounters at dusk, but could also be some pattern with more consistent calls at dusk. These relationships are hard to untangle, but the differences in frequency could be due to different behaviors at different times of day. So, what does this all mean? So, first of all, delphinid whistles were detected very frequently in this location. In some of the weeks I sampled, there were upwards of 1.5 million detections. So, dolphins are being very vocal around here. And so not only are there lots of delphinid vocalizations to pick up, passive acoustic monitoring does effectively capture these, so can be a really useful technique. We could have, this is just a very first foray, but we could have different delphinid species present at different times of years. Those really long, high-frequency encounters that we saw in the summer could potentially be species like common dolphins. They vocalize in this range, and they also are known to form superpods of many thousands of individuals, which could explain those really long, intense encounters as well. And then, as I mentioned, we also saw shorter, lower frequency encounters in the spring, and that could be species like pilot whales, which vocalize at these lower frequencies. Both these species were cited during the data collection for this study. So also saw differences in frequency throughout the day. This could be due to different foraging behaviors. So for example, not in Africa, but in Ireland, pilot whales have been observed to whistle more in the morning and forage more at dusk and night. So if they're, for example, whistling more during the day, that could explain those lower frequencies. But if they're clicking more at night, then you wouldn't see the whistles being picked up, so you have higher frequencies. However, knowing these patterns is important because it can aid in conservation. So, for example, I mentioned there were issues with bycatch. This could, if you know when certain cetaceans are foraging, you could identify sort of fisheries overlap and kind of introduce measures to reduce bycatch. But in order to do this, you will require species identification because, as I mentioned, none of these species are kind of confirmed. These are all speculative suggestions. So future research will require visual surveys to identify species in order to fully know their presence. Can also develop acoustic species classifiers, which some of my fellows from St Andrews are currently working on, but we're still quite a ways off. Also, this is kind of a really useful area to be doing research. There are clearly a lot of cetaceans here, and future research should involve local people, recruit candidates from local universities, and kind of have more funding for local researchers as well, because there has been a somewhat colonial sort of perspective on studying in Africa. So having more local people could sort of help increase local investment and conservation. Thank you everyone for listening. I've been May. If you have any questions, please feel free to ask, or you can email me at this address.
Thank you, May. Questions for May?
Hi, thank you. You double-check the detection from PalmGuard on the subset, is that right? Mm-hmm. Do you have an idea of the performances in terms of recall, precision, or accuracy? Or?
Not off the top of my head. Essentially, what I did was check a day from each week of data to see if it was missing any encounters, and all the days that I checked, it didn't miss any encounters. It might miss individual whistles, but the overall encounters were all picked up. I also, because I'd set the sensitivity quite high, because I didn't want to miss anything, it did mean there was a high percentage of false encounters. So I cross-checked, I think, 50 encounters and removed encounters with detections below 20 individual detections, because 85% of encounters with fewer than that were false. So I kind of didn't want potential false encounters skewing my data, and there were quite a lot of false encounters. I think I had something like 800 encounters initially, and after I'd removed the false ones, it was 365.
Okay, thank you.
So, yeah.
Any more questions from the audience? No? I also have one question. So are the dolphins, they usually stay near the shore for visual observations to be possible, or?
So for where these hydrophones were placed, this was 18 kilometers offshore, so this would require dedicated boat surveys in order to confirm. And there are potential, so for example, the Atlantic humpback dolphin that I mentioned, that's a coastal species, that doesn't really go that far offshore. So there are potentially slightly different communities in the two areas. So you can't kind of rely on onshore observations to know what's happening that far out. So yeah, that's kind of one of the things where it would be useful to have dedicated boat surveys, but there's no money.
Okay.
Yeah.
And when you made it verified, did you verify which species were vocalizing, or only like dolphin and that's it?
Yeah, only if it was, because there were a few anthropogenic sounds that were being picked up, I think, as dolphins, because they were tonal. But yeah, I only checked for dolphins and not dolphins because we just don't have enough data for this area to know what species are present. I mean, if you're experienced, you can recognize a few species. Like some of the behavior did look very much like kind of what I understand common what dolphin vocalizations to look like. There were also what looked like sperm whale clicks as well, but again, you kind of can't really be certain because there's not enough data on what the species in this particular area sound like. So,
okay.
Yeah.
Thank you, May.
Hi, I'm Camille Rondeau Saint-Jean. I would very much have liked to be there with you today in Brest for the Journée des jeunes bioacousticiens, but unfortunately I was unable to attend. I hope you will enjoy the video that I made instead for my presentation on how deep neural networks generalize across intraspecific variation using the example of Savannah Sparrow's song. So one might ask, why bother studying intraspecific variation? As biologists, we are often used to work and think at the level of species, but we know now that animal culture can be very important for the conservation of populations. In certain cases, cultural manifestations can act as a proxy for population structure and genetic diversity. But we have few tools to study intraspecific variation, and we don't know how existing tools handle it. We have many deep learning tools that work very well to classify animal sounds at the level of species. BirdNet is a very good example of that. It has been used for identifying bird species in very large audio datasets for research and education. So the question I wanted to answer was, how does a deep learning tool for species recognition like BirdNet handle intraspecific variation? In other words, does it have biases against certain cultural groups or individuals? To answer this question, we studied Savannah sparrows, which are widely distributed across North America. What is particular about this species is that males sing only one song for their entire life. That song is unique to them and can serve as a vocal signature. This makes them a simple, interesting case for our question. So here are songs from the same bird recorded at different moments. So you can see and hear that it stays virtually the same. Since Savannah sparrows learn their songs from each other, they can transmit cultural traits which are similar to accents in human languages and are called microdialects. In the population we studied, there are 7 microdialects, and we tell them apart by the middle section that I have put in colored boxes here. The differences are subtle and quick, but with some practice we can hear them. So this is microdialect P. Let's hear S. And let's hear W. But you can see that even though that section is very similar within the group, there is still some individuality in the song. For example, the final trill is different for all 3 Q birds here. But with all this, we wanted to investigate how a deep neural network like BirdNet handles intraspecific variation. In 2022, we went in the field to collect different Savannah sparrow songs to test if BirdNet has a similar performance on all of them. Fieldwork was led on Kent Island, which is a small island in the Bay of Fundy, right on the Atlantic border of Canada and Maine. It has a beautiful habitat for Savannah sparrow with shrubs and grassland and very few predators. First, we gave all Savannah sparrows color bands that will allow us to recognize them later with binoculars. Thanks to these bands, we made focal recordings where we know which individual is singing and get beautiful, clear recordings of their song. We collected around 90 hours of these. We also installed 30 autonomous recorders every 50 meters on the main study area. They recorded every day in May from dawn to dusk. So in total, we gathered over 15,000 hours of recordings. But since these microphones collect sounds from all around them, the recordings are much noisier, and you catch several birds at the same time and other species as well. I manually labeled an important part of these recordings at the level of the individual. In some cases, it was quite straightforward, but sometimes it was very challenging, as several individuals can sing at the same time. This, and I cannot overstate it, was long and tedious. Overall, I annotated just under 70,000 songs, and we estimate that there is less than 1.5% of errors in those labels. So in 2022, we recorded 51 individuals, but if you look at how they spread across microdialects, we had very bad class balance, despite all our efforts in the field to find and record more individuals belonging to the rare types. We tried to prioritize less common types when annotating, but there again, the class balance is not ideal. Still, it's a data set with which we can do a lot of work. Our question now is, when there are differences within the species, is a deep neural network able to ignore those differences and identify those songs as coming from the same species as opposed to other species? And does it give the same weight to all individuals and cultural groups, or are there biases? I wanted to investigate the performance of BirdNet on all these songs. Is every individual and microdialect of Savannah Sparrow as easy for it to recognize as a Savannah Sparrow song, or are there some types as easier than others? When we feed a song into BirdNet, this is what we obtain. It's a ranked list of species with a score from 0 to 1, expressing how confident the algorithm is that the species is present in the clip. So I have this kind of list 70,000 times. From all the clips, 81.9% of them had Savannah sparrows as the species with the highest confidence score. About 15% of predictions were for a species that was also present in the recordings, such as gulls, swallows, or Canada geese. In those cases, Savannah sparrow was also often present lower down the prediction list. Only 3.5% of recordings had a clearly incorrect species as the top prediction, which is quite impressive. However, if we look at the confidence score that was attributed to the species Savannah sparrow, no matter its rank on the list, we see important disparities between individuals. Note that the colors correspond to the microdialect, and we can't really see a tendency here. So some individuals that we see on the left here, like LGL and GWG, receive very low confidence scores, whereas someone like RYB on the right consistently gets high scores. Is this due to characteristics of their songs? Maybe, but we suspected that the effect we were observing was due to the fact that Savannah sparrows like to sing from very few perches over a small territory, and they prioritize those perches over the whole season. And since some sing very close to the microphones or directly on them, they get very loud, clear songs that are easily identifiable for the algorithm. Some others always sing far from any microphone, therefore their songs are faint and overlapped with various noises. To mitigate this bias, we decided to parse through the focal recordings and for each individual choose 8 songs that are as clear as possible. We were hoping that this would put all individuals on the same level of comparison. If the algorithm doesn't discriminate on cultural factors, we then expected to see all individuals perform equally. However, we had quite a surprise. On average, the scores are much higher, as one would expect from high-quality songs. But again, there are huge differences in the scores received by different individuals. Some get almost perfect scores, and some have much larger variance, including surprisingly low scores, once again on the left side of the board. When grouping these clean clips by microdialects, we can see a similar phenomenon. Although the classes are quite uneven, with much more individuals in the P and T categories we see in the middle, we see that intriguingly, the S type, for example, tended to get low scores. On the contrary, the D type on the right almost always gets perfect scores. But why is that? Is it again due to some biases we weren't able to eliminate, or is there something in the structure of the songs that the algorithms react to differently? Is it maybe due to an imbalance in the data set used to train BirdNet in the first place? Those questions are still in the air. In conclusion, we've seen that BirdNet shows a good performance in that it produces a very good proportion of correct or plausible predictions. However, we've also shown that some individuals or cultural differences may lead to uneven results, and that needs to be addressed. Future research should investigate why we observe such disparities. Is it due to certain song types being less represented in the training data? And how can we mitigate for this? Also, we've observed that phenomenon at a very small scale over the island of Kent Island. But does it also exist at the regional or continental scale? How does that affect the validity of results obtained from large-scale surveys? Finally, you've seen that we've constituted a large dataset with detailed annotation, and the obvious next step is to experiment with training algorithms for individual and cultural recognition. And we will be happy to share more when we do such experiments. Thank you very much for listening. I hope that you have a great time in Brest, and don't hesitate to write to me with my email address here if you have questions or wish to discuss my work further. Thank you.
C'est bon? Up next is Charlie, who's going to be talking to us about bottlenose dolphins. The floor is yours.
Alrighty. Hi everybody, I'm Charlie. I work at the University of St Andrews in Scotland, and I'm here to present some of my work on bottlenose dolphins and geographic variation. So, first off, what do I mean when I'm talking about geographic variation? So in this context, we're referring specifically to variation in the acoustic parameters of calls within a single species and across geographic locations. So this is something that has been demonstrated in several taxa before, from frogs and insects to birds, cows and dogs. It's pretty ubiquitous. And this is important to take into consideration, not least because the Convention on Migratory Species recognizes vocal identity as relevant to culture and therefore biodiversity, stock assessments and conservation, but also, and more importantly for our interests, it's really important to take into account when we're looking at species classification. So, allow me to introduce you to ROCA, the real-time odontocetes call classification algorithm. So this is a program, it's a module for PAMGuard that was developed by Julie Oswald and others. And it's essentially a set of machine learning algorithms, which is very difficult to say apparently, which is used to classify individual encounters and vocalizations to the species level for odontocetes. So they require development for their region of use, depending on the various species you have in each ocean basin. But at the moment, we're somewhat uninformed as to over what geographic ranges they can be used. So here we have a little screenshot from ROCA. Essentially, we've traced the frequency contour of a whistle, and this will spit out several acoustic parameters for us, a total of 46. Before you ask, no, we are not sponsored by McDo. But so yes, we have to take this variation into account when we're then trying to train any species classifiers so that we know over what ranges they can be used and how effective they might be when we're using them in different areas of the world. So geographic variation has been analyzed in this species before, but much previous study has focused either on broader call categories, so human-assigned classifications, general shapes, things like upsweeps, downsweeps, arch shapes, that kind of thing, which is perfectly valid and fine. But one thing that few of these studies do take into account is a unique vocal habit of the bottlenose dolphin, and that is the signature whistle. So these are individually distinctive and highly stereotyped signals. And the problem is that bottlenose dolphins do it a lot. So 38 to 70% of the repertoire of free-ranging dolphins can be made up of signature whistles. On the side here, you can see an example of some of the ones that we record down in Sarasota in Florida. And you can see they're very diverse signals, but each one is unique to an individual. And this has the potential to hugely skew our data set or any classification towards the individual level rather than a population that we're interested in. And also, we don't always have a nice, neat catalog like this. So where Rosie, you heard from my colleague earlier, was presenting on signals that had been recorded from a hydrophone on the melon, we can't always do that. So in that case, when we have wild populations, and I don't know what's gone on with the slides at all, we have developed a method for identifying them in free-ranging dolphins. So this is known as SIG-ID. I know you can't quite read that, but it is there. Whereby 4 or more stereotyped whistles that are found within 1 to 10 seconds of another are likely signature whistles. And this is a very conservative set of parameters, meaning that we have a very low false positive rate when we use them. So, for this study, we have 3 main research questions. Firstly, for any analysis of geographic variation to be relevant, we need to know whether it exceeds the variation within a site, so inter-individual variation. Secondly, and probably most obviously, can we distinguish between geographic locations using acoustic parameters alone? And lastly, does this remain the case when we have populations whose ranges overlap? So, for this study, we had 7 sites across the Northeast Atlantic, the Gulf of Mexico, and the Eastern Tropical Pacific. And there we go. And the data were recorded from a variety of sources and organizations. So we have a mixture of focal follow and line transect surveys. We have several different hydrophone arrays in use. The range of frequency responses is given there. If anyone wants more of the gory, nerdy details, by all means come and talk to me later. But the most important thing is all of these were recorded in conjunction with visually confirmed sightings. So we have species ID, and we know all of these recordings come from bottlenose dolphins. After this, we then manually extracted the various features in Rocker, which I was showing you before, and we used these 46 parameters to train a series of random forest classification models. And again, this has been chopped off, and I'm not quite sure why. Luckily, Angela explained some of this for me earlier. But these are essentially a very basic form of machine learning, whereby various decision trees vote for the class to which they would assign a sample. Given we're talking about random forests, I went with the most random forest I could think of to represent them. So here's Treebeard. So returning to signature whistles for a moment, we needed to control for their presence to avoid this skew towards the individual. So firstly, you can see on the top left there, we have an example SIG-ID bout. So we have this repeated signal, which is likely a signature whistle. And what we did was take only the first example of each. So in those red boxes, those are the whistles that we then took out, the first example of a bout. Secondly, for each encounter, so for each recording we have with bottlenose dolphins, we then ensured that only 5% of the data, of the 5% of the whistles in that encounter were signature whistles, so that we weren't bloating it with this highly stereotyped signal. Once we'd done that, we could move on to the classification. So, we had 3 primary model configurations for our 3 questions. Firstly, our first random forest was trying to classify within the East Scotland population. So here we have a resident population, and we are trying to classify to acoustic encounter. So we have complementary photo ID for this site, which meant we could ensure that there are no individuals present in multiple encounters, and therefore remove some. We knew that we were classifying between distinct individuals. That's the point there. There's something gone very wrong with the formatting on this presentation. I'm very sorry. Our second random forest was then trying to classify to geographic location, so to one of our 7 sites. The keen-eyed among you will notice that there's 6 classes rather than 7. We had to eliminate the Bay of Biscay because we didn't have enough whistles. And then lastly, our third configuration attempted to classify between 2 adjacent populations. So we have the east coast of Scotland and the west coast of Scotland, where we have some known transfer of individuals, to see whether it's still possible to classify between them. So for each of these model configurations, we assessed those 46 variables for collinearity using Pearson's tests, and removed any highly collinear variables. We then randomly sampled from each encounter, split those into training and testing sets. The splits varied from 70 to 80% depending on the configuration in question, and then trained and tested our random forest models. This process was repeated over 100 iterations, And from that, we could then take the mean diagnostic variables, so your mean accuracies, your Cohen's kappa statistic, which Angela was explaining before. It's an inter-observer reliability test, basically. But in this case, the observers are the model and us. So, what did we find? This is a confusion matrix. So essentially, on the y-axis, you've got the true classes that they've come from. On the x are predicted classes from the model. And within each cell, you have a percentage. So basically, the lighter the color, the more accurate it has been. So from what you can quickly tell from that confusion matrix is the first model is rubbish, completely useless. So we cannot classify to encounter. Mean accuracy of 28%, it's a terrible classifier. But then, when we move on to the second one, where we're looking between geographic locations, all of a sudden we have a nice, neat diagonal. Mean accuracy of 71%. Cohen's kappa statistic, again, as I've explained earlier, significantly improved. One thing you get from a random forest analysis is then an assessment of the Gini index, which is essentially variable importance. It's how useful it found it, the model found it to classifying. So we took the top 4 of these and ran a series of Kruskal-Wallis analyses to see how and where these calls are differing parametrically. So one example then, when we're looking at the percentage of flat whistle contour, so how much of the whistle was horizontal on the spectrogram. This was significantly lower in East Scotland and the Eastern Tropical Pacific as compared to the other sites. Similarly, Hebridean dolphins had a much lower frequency, a significantly lower frequency in the middle of the whistle, at the mid-sample. And Welsh whistles were significantly longer in duration than their UK counterparts. As a Welshman myself, this makes a lot of sense. We do like to talk. So that answers the first 2 of our questions. We do indeed have greater inter-site variation than within a site, and also we can reliably classify to geographic location. We move on to our third model, where we're looking at our overlapping populations. So we have a resident population in Scotland in the Moray Firth, which is that little divot you can see at the top on the right. And though this is thought to be resident, we do have some documented cases of dolphins leaving that area. So one keen example is Spurtle, the sunburnt dolphin, who has been recorded off of southern Ireland and also as far away as the Netherlands and Denmark. So there is definitely transfer of individuals out of this area and across into adjacent populations. This model also performed very well, in fact the best of the lot, with a mean accuracy of 81%. So we can reliably classify between these 2 adjacent populations where we have transfer of individuals. Similarly, we looked at some of the variable comparisons between these. They're actually very similar variables to the second model configuration, so it seems both configurations were picking up on similar traits within dolphin whistles. So this answers our third question. It does indeed remain the case when populations overlap. So, in summary, whistles are varying measurably between different geographic locations. We're exceeding intra-site variation, and it's present even when we have adjacent populations. Then this has highlighted some areas where our species classifiers might find some difficulty. So especially when we're looking between East and West Scotland, we might need to tweak some of our existing classifiers for the Northeast Atlantic, or even better, develop separate classifiers for each one. Moving forwards, we'd obviously like to include more vocalizations, so clicks and burst pulses, the rest of the dolphin repertoire. We'd also like to include more sites. I think one of my supervisors is famous for saying she always aims for world domination, but we'll get there one day. We'd also like to do this with the other species that are involved in all of our classifiers. We have several other delphinids to do this with. And also looking into some of the drivers of this variation, so whether there's any behavioral reasons for these changes, or whether we're looking at dialect level differences, social splits, that kind of thing. So I'd like to thank all of my collaborators back in St. Andrews, all the institutions that provided all of our data, my supervisors, and my co-workers. And yeah, thank you very much. Any questions?
Thanks, Charlie. Have you any question? I think they're tired. Okay. Thanks.
Sure thing, no problem.
Up next and final talk of the day, grazie mille, is Lia Zampa, who's going to be talking about the strength of acoustic identity coding in territorial bird species.
No, I'm better like this. No, I will keep it. I move a lot. Just Okay, so hello to everybody. I'm Lia, and we are going to keep going with the individual vocal signature, but in territorial bird species this time. I'm going to present some of the results of my PhD. So, the reason why being distinct from the others in nature is important is still debated. Of course, we know that being recognizable is crucial for tasks with an important effect on fitness, such as parent-offspring interaction, the maintenance of complex hierarchies, or the maintenance of cooperative breeding system, for partner selection, resolving sexual conflicts. But so far, the selection for individuality is thought to be particularly strong in species with large and complex social groups. But let's not overlook two territorial species that have their own unique distinctive signals, especially in calls or songs that are related to territorial defense and mate attractions. In fact, a correct discrimination between rivals may prevent unnecessary and costly aggression. I know you, you're my neighbor, you're not a threat, you have your own territory, you're female, so I'm not going to spend energy to fight with you. So the role that intrasexual selection have on shaping these vocal signals can be a determinant, and we want to dig more in this role, asking two simple questions. If the breeding density have an effect on the individuality of vocalization in territorial bird species, we assume that in a more crowded area where the pool of potential rivals is higher, the male should be more distinctive. So the overall vocal identity should be higher because they might want to sound as different as possible from the other, compared to area where there are less male, and so the competition for the acoustic space is simply lower. And consequently, even at individual level, to avoid error, mistake during the recognition process, we assume that neighbors should be more dissimilar than farther individuals. So we are going to test these two simple questions. We tested these two simple questions in two completely different territorial species, different in habitat, song production, habitat, song production, and phylogeny. Okay, the first one is pretty famous if you Google it in French cuisine. Apparently it's a delicatessen, is ortolan bunting. It's a migratory species from Eurasia. His song is pretty simple. The male is composed in two parts, this initial and final phrase that differs in the overall frequency and bandwidth. Males within the same dialect share this final phrase, while they have kind of two or like maximum up to four different initial parts that are like characterized by the repetition of the same syllable type. For example, here is the repertoire of a single individual who sings these three different song types. So we sample population breeding four different sites in Wielkopolska region in the northeast of Poland that are characterized by different habitat fragmentation, and in which we can distinguish sites like cluster with increased number of the individual within 500 meters, that is the hearing range, basically, of ortolans, and a really small cluster with just 3 up to 4 individuals. These are the colors of 2 different here. And then we move to another continent, and we studied emerald spotted wood doves who breed in savanna woodlands. Even here, we can see that the song is characterized by 2 parts. We have like this really soft initial phrase that have notes more modulated in frequency, that it's contrasting with the really loud final phrase with the unmodulated notes. We sample, we recorded and follow a population that breeds in Lake Umbura National Park in Uganda, and here in a more bushy area with denser vegetation, there are much more individuals respect to more open space with scattered trees. So there we have our high density and low density area of males. And so what... Okay, so at the beginning of the breeding season, when the birds are vocalizing a lot to attract the females, we start the data collection. We record as much birds as possible. We capture them. We ring them, and then we, with some software, we extract frequency and time domain parameters of the songs. And then we first calculate this Beecher statistic to compare between different groups of high and low density population. This is the best available metrics to measure within group signature, and it's basically higher is these values, this Beecher statistic, easier is to discriminate the male within these groups. So they are more distinctive vocally, basically higher is this value. And then to explore the spatial variation of this individual signature, we calculate some pairwise dissimilarity scores between pairs of males. For Turtur, we use the matrix of result from a dynamic time warping using Lucinia. And then for ortolans, since they have different song types in their repertoire, first we calculate the acoustic space of each individual, and then we measure the overlapping of the space between pairs of individual as a measure of similarity, basically, of their songs. So what we found, that ortolans have pretty individual distinctive songs. Here are 13 individuals who sing the same C1 song types, and we can see from the PCA and from the Beecher statistics that both initial and interesting in this final phrase, and is thought to be like a signal of dialect recognition, are pretty individual distinctives. And even if you look at the more understudied non-learning species like these doves, they have in both components of the songs pretty strong individual signature. Okay, so they have a distinctive voice, but what about our research question? Is it stronger in more crowded area? So, if we compare the Beecher statistic of the 3 closest male that lives in a crowded area and no crowded area for ortolan bunting, we can see that in 2022, it's pretty obvious and it's significant, the difference between the 2 groups. So male belonging to more crowded area have more distinctive voice respect to the one that lives in just low, that are just 3 isolated male. In 2023, the difference is not significant. We will see what 2024 will tell us. For Turtu, interestingly, the situation change based on the type of song phrase that we are taking into account. Because if we look at the final phrase, we can see that in high density area, males are much more distinctive in this. They have like more. They are more discriminable individually in this final phrase, and it kind of makes sense if you look at the picture of this high-density area. The signal of the information of the identity need to reach more males and need to go through this huge amount of bushes. So it makes more sense that I put the effort more on the final phrase. While in the situation is the contrary on the initial phrase, that it seems to be more distinctive for in open space. But then this result may lead to other questions if like the initial and final phrase have actually different function. Maybe the initial phrase is more like to attract the females, while the final phrase have more like a function of territorial defense. But more will be done. But if we look at our second question, if the neighbors are more similar than no neighbors, our expectation were not actually covered, because the closest male are for ortolans. They are more similar, like neighbors are more similar respect to known neighbors. So besides being a byproduct of the vocal learning, being like having this group signature of neighborhood may have even some benefits in case of an intrusion of foreign mares from another side, or some floaters that are pretty common in ortolans, since there are not so many females. For what concerns the tutor about this result, there is no significant difference in neighbors and known neighbors regarding like the similarity of their songs. We can see a trend, but I'm still working on this model, which suggests that even here, like closer male are more similar respect to farther one. So to sum up, we verify that even in the territorial species, there is a large potential for identity transmission in their songs. There is the support of the role of intrasexual selection as a driving force for acoustic individuality, because it seems that there are differences in more and more crowded and less crowded area. But besides being individual distinctive, there can be an interplay between the individual recognition and maybe a group recognition, as they suggest by the second part of the results. And then now we need to even think about the importance of the detection radius of the identity information, at least in Turtur Carpospilo. Maybe even the environment can lead a role in where and how to shape even the signature in the species. So I would like to thank all the collaborators, African ones, Polish one, and Czech the team for the help and the support. And I'm still working on other stuff, but I would be happy to receive questions, suggestions, and thanks for the attention.
So, any question?
Why do you think there is a change between years in the identity?
So, the brutal truth is, I think that there is a bit of bias in the sampling, okay? Because in some sites, we do arrive too late in some big cluster, and a lot of birds were already paired, and we didn't manage to get all the individual, okay, all the neighbors, and I think that makes a lot of difference. I mean, we have like few songs, but they're not enough to extract parameters. So I think that we really need to have this year as well, that we just finished to record them, and maybe we can get other data from previous year. But that's my main guess. And then actually ortolans are pretty difficult, because they're kind of decreasing a lot of so every year there are less and less, and I think that there can be like a lot of moving between sites. we have recordings, and I took into account in the analysis the individuals that are stable. They're okay for all the season. We individually mark them, so I'm sure that that can be. But yeah, I think that can change a lot, even between years. They are not so stable in the coming back there, I would say. So that can be another problem for testing this hypothesis. But yeah, bias and maybe the mother species, that it's a bit tricky.
Yes, thank you. It was
No,
a really
okay.
nice answer.
Yeah, thanks.
Another question?
Do you have like fitness data by any? Sorry, I guess that's.
No, sorry. No,
Ah,
no.
okay. No, because I guess like maybe if you have like a dissimilarity or beach index by individual, maybe, yeah, maybe like more dissimilar individual have like, I don't know, better fitness or something like that. So
Yeah.
maybe you could link it to it.
So no, unfortunately, we don't we didn't have the possibility to check like even the nest, even because it's super tricky. It's a kind of shy species. Is difficult because then they breed into the crops. They have the nest into the crops, and yeah, and since they are even in danger, like it's hard to get even the permits to study them there, and it's for a time constraint like point of view because we also need to go to the tour in Africa, and so we don't have time to follow all the season. Basically, we need them at just the beginning, but it would be interesting. I mean, there are some papers who said who actually analyze fitness-related traits with the repeatability of the song and the consistency of the song, and they see that the more consistent the one that they can match and copy better, like their songs, okay, and do it in the same way more time, the same bout, there is low variation, they have actually better fitness, and they are more chosen by the females. It was, I think, with pie flycatcher or great eats. Recent, it just went out in Science or Lower Nature. So I think that it's, yeah, they kind of like stability. Okay, if a male can copy better, like its own song and do more matching, it's kind of a signal of quality. But it's even true that I think that it's a trait that is species dependent. I wouldn't say that it's the same for all the species, I would say. Yeah.
And I have a second question.
Go.
And also for when you plot like the dissimilarity across the distance, did you do like categories of distance, or it was like really the actual distance? Because sometimes when you play a little bit with like some distance, I mean, with many species you have like you know, that kind of shape of curve.
Yeah.
Depending, on your categories.
Yes.
And it's true that we always thought, I mean, with my supervisor, we are always saying that represent the dispersion, that you're really close neighbors that are not necessarily like really similar to you, but a bit further away, like just the second category, like just 100 meters away, for example. They are like really, really similar, but in a way, like, with your interpretation of things, it might also be that if they're really close to the neighbors, they should be a bit dissimilar. So I wanted to know if
I do try with the distance category every 100 meters of distance, basically, and I would say that that's a bit. The pattern is the same, okay? You have a kind of, sometimes it's even more tricky because you have like a peak of 300 meters and then it drops down, but it wouldn't make sense why it's like at the closest individual like are more like similar, okay? Then the one of 300 meters are much more dissimilar, and then they keep going to be more similar. So it didn't work well for the categories for me, but it's even true that I have a lot of different way to calculate this dissimilarity, and to be honest, it's kind of different. So now I'm just struggling even for that because is different, this pattern, as you take into account different kind of variable, how you calculate it. I use the overlapping of acoustic space because it was my final, okay, it's biological, makes sense, okay? But if I take just the initial and final phrase, I will have a bit of slightly different result and different pattern, but they are not significant. So yeah, I'm a bit struggling with the, with the data resolution, but thanks for asking. Yeah, we can talk later.
Okay, thanks, Lia.
Okay, no.
Okay, so we'll see you in 2 hours' time for the reception gala dinner. But also just a quick note to say that our time has come and that most, if not all, of the organizing committee will be leaving the committee itself in the following month. And it's been an amazing experience. It's been really fun to build this event. So if any of you guys are interested in taking over next year or the year after that, please feel free to come to us or chat with each other to see who's motivated. And so we can find a new organizing committee for next year. Have fun during your free time.